Both abiotic and biotic factors influence secondary succession. They are essential to comprehending landscapes altered by human activity. In low-stress environments, competition and consumption play a larger role in secondary succession. Herbivory during succession selects for unpalatable plants, which can lead to the rise of non-native species when native competitors are consumed. However, it can also inhibit non-native species if they are consumed. Estuaries respond to stress in a variety of ways. To gain further insight on the effect of herbivory and environmental stress induced on coastal marsh vegetation during secondary succession, we study the community structure along with the rate of plant recovery in artificial bare patches in Narragansett Bay. In these estuarine marsh patches with differing salinities, small mammal herbivore pressure was shaped experimentally. Prior research leads us to believe that marshes with lower salinity will exhibit that the relative importance of small mammal consumers in the driving pace and species composition of secondary succession will be greater in this environment. We disputed whether the role of herbivores on secondary succession differed from their role in undisturbed environments.
We identified salt marsh sites within the Nag Creek system, and brackish and Aligo-Howline marsh sites were located along the Barrington River. We utilized 25 by 1 meter transects in each type of marsh, with bare patches smaller than 0.1 meters squared not being included. In Aligo-Howline and brackish marshes, seedling-only areas were included. In salt marshes, large hypersaline bare areas were excluded, and only areas that showed evidence of recently killed vegetation were used. We performed a patch recovery experiment with locations that varied in salinity stresses and mammalian consumer pressure. There were 3 variations in bare patches: cage, cage control, and control. Cages were made of mesh that excluded ground access to rodents. Cage controls were similar in design, with the bottom edge being raised 0.25 meters to allow ground access. We quantified species cover bi-weekly for the first 6 weeks of the experiment using a point intercept method. Recovery was measured at the end and beginning of the growing season for 3 years, from 2005 to 2007.
In order to test if rodent herbivory effects were similar in invading plants and in recovering plants, consumer pressure was manipulated in mature stands of Phragmites australis, an invasive species in New England. 30 half-meter by half-meter plots were located along 200 meters of shoreline. We established plots along the boundary of a dense P. australis stand where P. australis was expanding into a mix of S. patens and J. gerardi. A similar cage control method was used. Cage controls had 2 0.3 by 0.1 meter holes cut in the bottom. Within each plot, stems of P. australis were counted at the beginning and end of each growing season. We found rack-generated bare space was present in all marshes and varied with salinity. Oligohaline marshes exhibited less bare space than both brackish and salt marshes. Secondary succession speed was affected by the interaction between herbivory and salinity. Herbivory impacted recovery in brackish and oligohaline marshes. Salinity stress also slowed secondary succession. Salinity and herbivory both affected species composition in recovered communities. Herbivory lowered species diversity. Species varied in response to herbivory and salinity. Some species were significantly affected by both, while others were only affected by one. Herbivory displayed a diminishing role in species composition from low to high salinity. Rodents did not control the growth of P. australis.
Our research indicates that small mammals fill a keystone role in the New England marsh ecosystem. Physical stressors, such as salinity, play a dominant role in recovery of disturbance plots. When physical stressors decrease in intensity, small mammal herbivores have more influence on the community structure and recovery rate. This change from bottom-up control by physical forces to top-down control by herbivorous consumers independently parallels similar scenarios where human actions have initiated consumer control of marsh plants without evidence of human influence. Herbivores play a large role in controlling the establishment of invasive marsh grass in bare or disturbed areas.